Hello guys, welcome back to another science video from the Howard. Today we're looking at photosynthesis, and in particular we're looking at how light intensity affects the rate of photosynthesis, and that is chapter 8 in Trilogy. Now what we have here is we're going to have some pondweed. Now pondweed is a green plant, so that means photosynthesis is going to occur in our pondweed. Now we've come across photosynthesis in your studies in the class, and that is simply when we've got carbon dioxide reacting with water to produce glucose and oxygen. Now with sunlight and obviously the chlorophyll, which are in our green plant cells here. Think about also linking this together about what you know about the cells in a plant and the organelles in there, and what organelle in particular we are focusing on here if we're looking at photosynthesis. We're looking at the chloroplasts. Now the setup here, I'm going to show you two different options to do. First of all, we're going to have a look at a way of doing this practical within a boiling tube. And the second option I'm going to show you is by using an inverted, upturned funnel with a measuring cylinder on the top which has been filled with water. We'll get to that shortly though. So what I'm looking at here is I'm going to take a piece of pondweed. Now this is a particularly good plant to look at if we're looking at photosynthesis and the production of oxygen. And what I'm going to do to start with is I'm going to put it into a boiling tube here. Now I've filled the boiling tube almost up to the top. So we want this pondweed to be submerged. It's really important that it's submerged, and particularly the top here, the stem here, we need to make sure that is pointing at the top. And we're actually going to cut it as well. It's been freshly cut, but I'm going to cut it again. So I'm going to submerge it in here, pop it all the way down, push that in. So you can see here that the top here is submerged below the waterline, but I'm just going to cut that again with some sharp scissors. Okay, there we go. Now, to get this going, we're actually going to turn our light on here. We've got LED light. Now, it's important that we think about the variables we've got here. Now, I'm going to turn this on so it acclimatises to that solution. And then we can then study the rate of reaction. We'll leave that for about 5 minutes for it to acclimatise. What we're looking at here is light intensity. That is our independent variable. It's the thing we are changing. And how we're doing that is actually by changing the distance we've got from the lamp here. And we're going to move it along in 10 centimetre increments. So I've got it positioned at the moment at 10 centimetres. Now the things we need to keep the same. Well, we're changing light intensity, so we need to keep the temperature the same. That's why we're using an LED bulb here. So we want to keep the temperature as close to the same for each experiment as possible. If this is giving out a lot of heat, then obviously when we're closer to it, the temperature is going to change, and therefore that's going to affect our results as well as just the light intensity. The other thing we're keeping the same here, another control variable, is our concentration of sodium hydrogen carbonate. If we altered that, then the percentage of carbon dioxide within that solution will also change, which could then also impact the rate of photosynthesis and therefore the production of oxygen. So we must keep those the same. Right, so now I've left my pondweed to acclimatise to the water, just about 5 minutes now. We've got a steady stream of bubbles of oxygen coming here. Now what I'm going to look at now is recording and counting the number of oxygen bubbles produced over a time period. Now I'm going to do this for a set distance from the bulb. I'm going to measure 10 centimetres from the bulb, from the front of the bulb to the centre of our boiling tube here. And then I'm going to repeat that 2 more times to get 3 results, and I'll then take a mean. I'll then move our boiling tube along to 20 centimetres, then to 30, to 40, to 50. I will then be able to plot a graph of volume of oxygen produced against distance from the bulb. Now the time period I'm going to use for this, the production of oxygen in this particular boiling tube is relatively slow. So I'm actually going to do it for 5 minutes. It will give us a better set of results if we do it over a longer duration, especially as we're going further and further away from our bulb. So I'm going to hit the stopwatch, I'm going to count the oxygen bubbles, and then we'll get our graph and table of results started. Right, now the other way I was telling you you could do this is if using an upside down funnel and then using a measuring cylinder. So just to demonstrate what this would look like, I have a large beaker here, very large beaker of our sodium hydrogen carbonate, and I have an upturned funnel. It's a normal funnel, we obviously usually using this way up, but I'm going to turn it upside down so that it can then channel the oxygen bubbles and then send them up through the spout of the funnel. And then I have a measuring cylinder which is absolutely full to the brim with our sodium hydrocarbonate solution. All I'm going to do is take another piece of pondweed, so I'm going to select a nice piece here, and I'm going to put it inside the inverted funnel. I'm just again going to just snip off the end with a sharp pair of scissors, and then I'm going to place it inside our funnel with the bit I've just cut facing upwards, and I need to carefully place this into our beaker and lower down. There we go. Now the next step I need to do is a little bit tricky, because I need to get the measuring cylinder, which is full of our solution, inverted without losing any of our solution here. So I'm going to put my finger over the top, I'm going to invert it, place it into our solution, and then carefully place over the top of our funnel and then lower down. Now what that is going to enable me to do is actually to record a volume. Rather than counting the number of bubbles, I could then record a volume. So it's the same process. I could then set my lamp up at the edge of the ruler here. I could then move my beaker to set increments, so it could be 10 centimetres again to start with. Again, the point we're measuring needs to be the same. So again, it needs to be from, if I'm going to choose the end of the bulb, and then I'm going to choose the fact that I'm going to go for the middle of my funnel, so right where the spout is, and then get it in line, lower the lamp down, measure up, and then turn on. Again, I'm going to leave this for a while, at least 5 minutes, possibly longer, to get it acclimatised, and then I would start doing my recordings just as it would be before, so I could use a 5-minute time interval. Again, this depends on the concentration of your sodium hydrocarbonate. The more concentrated that solution is, the greater production of oxygen is going to be, because that is a factor that's going to affect the rate of photosynthesis. I'll then pick our distance increments again, and then record 3 results for each distance. And then again, record them in a table, and then plot our graph, which I will show you how to plot in a moment. So this is all about variables, really, and making sure that they are very, very clear. Your independent variable is the light intensity, which you're changing by using distance from a lamp. We're using an LED bulb here to reduce the amount of heat production from our bulb. Temperature is going to stay constant, it's a control variable. The concentration of the sodium hydrogen carbonate solution is going to stay constant, it's a control variable. Also, you're going to make sure you use the same piece of pondweed each time, so that pondweed will stay in there the whole duration of the practical. These must be noted as control variables. Our dependent variable is either the number of oxygen bubbles produced in a time period, or it's the volume of oxygen produced in a time period. Let's move on to plotting our graph, and I'll talk about how we can look at these results.
Okay, so I've plotted my graph on Excel for this one. On the x-axis we have the distance from the light source in centimetres, and on the y-axis we have the rate of bubble production over 5 minutes. So this is the rate of oxygen production, this graph, and we can see at 10 centimetres we produce the highest number of bubbles, and this decreased going further away from the light source. Now we may have expected to be a straight line here, and as the distance doubled, the number of bubbles halved. But that's not what we see when we do this practical. What we actually see is, for example, from 10 centimetres to 20 centimetres, although the distance has doubled, the actual, approximately on this graph, the number of bubbles produced is actually divided by 4. And that's a trend we see further down as well. If we go from, for example, 20 centimetres to 40 centimetres, I believe at 20 we produced 17 bubbles, we actually went down to 5 bubbles for 40 centimetres. So again, although the distance has doubled, the number of bubbles produced is not divided by 2, it's actually it's closer to dividing it by 4. Now this is what is called an inverse square law. Now the reason for this is as the distance goes further, the light goes further away from the plant, the light gets spread out over a factor of 4. So this is why we see this curved graph here, not a straight line. So just be careful when plotting these graphs. You do make sure that you're plotting your variables on the right axes. We always plot the dependent variable on the y-axis, and we always plot the independent variable, which is the thing we are changing, on the x-axis.